Development of the culturing method for the viruses. There are different methods to culture the virus in the laboratory. Of course, we need the viruses for different point of views. We need the viruses most of the time. We need them for the to see them, to study them for different purposes. Just like we can need to prepare the vaccines, we need to diagnose some specific kind of the virus, or we need some other reasons for study. We culture them. So there are different methods which are derived for the viruses to culture them inside the laboratory. First, cell line. There are different kinds of the cell lines which are used to culture the viruses inside the laboratory. First of all, the cell line was developed in 1912 by the Carroll and his co-workers. And first cell line also won the Nobel Prize for its culturing of the viruses and the cell lines. This was the fragment explained in the chicken embryos, and the heart of the chicken embryo was used to develop this cell line. And it was maintained by washing with the Ringer solution. So for the first cell line, which was developed in 1912 in the chicken embryo, and it was used for culturing of different viruses. Until now, we use chicken embryo for a lot of culturing of the viruses, and this was very important from the culturing point of view. Then developed in 1940 and the 50, there were larger number of the cell lines will be developed, and then the John Franklin Anders, Thomas Hucker Weller, and then the Frederick Chapman Robbins, they developed us different kind of the cell lines. We will see the cell lines in detail. First culture when they received the Nobel Prize, because of this was the very important thing, that to culture the virus, and then culturing viruses in the monkey kidney cells, and then they won the Nobel Prize for this. Early use was naturally derived product from the serum. The media was used, and these are the media which was used for culturing the viruses was the naturally derived product from the serum of the blood of the human beings or the animals. Later, chemical based synthetic media was ready to use. So first of the time when the cell lines or the culturing started, we used the serum, and then chemically derived different kind of the chemicals can be synthetically, the media can be used just like LB or just like the other things. Observation. Using the modern microscopy techniques, we can observe the different functions or different the viruses, and for the what specific purpose we are going to study the virus, we can see them using the microscopy techniques. And then the possibility to observe the inclusion in the cells and as a result of the virus. So this is one of the thing which we can see under the microscope. The inclusion bodies are the different life cycle of the virus, or what is the virus is causing inside the cell. So for this purpose we can use the cell culture. So possibility to observe the inclusions in the cell as a result of the virus, as a result of the infection caused by virus, we can see. And we can also observe different kind because of the microscope, different other things, just like the mechanism, how the virus can cause the disease and how the virus will be the culture here. Cleavage stage embryo. First of all, if we take the example of the embryo, that chicken embryo, that how the chicken embryo cause the disease, and how the chicken embryo, the cells are, the viruses are cultured in the chicken embryo. First of all, the chicken embryo is stage and it is cleavage so that it's clear, and then the culture the biocyst, blast cyst in it is then kind of the virus, then isolated the inner mass. Then the virus will cause the infection, and we can after the infection we can isolate the inner mass of that, and isolate that inner mass, and then that inner mass is caused and put in the petri dish, and then cells are disassociated and replated. The cells will cause in the petri dishes, then they will be the dissociated, and then irradiated mouse fibrillary the feeder cells. Then the new feeder cells will be taken, they will be dissociated from those cells, and they will be caused in another petri dish, and then the established embryonic stem cells cell cultures. So this is just an example of the cell culture, how we can do the cell culture method. So then there are different kind of the cell lines which are available, and which we will use them these days for the culturing of the these are L cell line, these are the HeLa cell line, these are some other cell lines. We will see them one by one. The L cell line was developed by Early in 1948. It was developed from the subcutaneous mouse tissue, and here you can see that how we use them and why we call them as a L cell line. It is clear from the picture given side below. Then the HeLa cell lines. It was developed in about 1950 to 1960s, and these are the diploid cell lines. These are developed from the human tissues, from the cervix, cervical of the human tissues. We will see them in detail. And then the Vero cell lines. These are also developed in about 1950 to 60, and these are the diploid cell lines, and from the simian tissue. See them in detail that what time that these cell lines have been originated. First of all, the L cell lines, we will take the example of L929. These are the mouse connective tissues. These are the fibroblasts. From morphological point of view, these are the fibroblasts, and they were developed in 1948. Then the HeLa cell lines, as we already see that these are the most of the cell lines were developed in between the 1950s to 1960s. Similarly, the HeLa cell lines are developed from the human cervix. These are the epithelial-like structures, morphological point of view, and developed in 1951. Then the CHO, which is abbreviation for the Chinese hamster ovary. Of course, these are from the Chinese hamster, and epithelial from the morphological point of view, and these were developed in 1957. Then there is the MDCK cell line, which were developed from the canine kidney, and morphological point of view, these are also the epithelial cells and developed in 1958. WI38, these are developed from the human lung, and these are the fibroblasts from the morphological point of view, developed in about 1960. Then BHK, Syrian hamster kidney cell line, fibroblast from the morphological point of view, and developed in 1961. Vero cell lines, a very common cell lines, were developed from the African green monkey kidney, and these are the epithelial from the morphological point of view, and they were developed in 1962. So from here you can see the cell lines have been developed from a large number of the animals and even from the human beings. We can develop the cell lines from the mouse, from Chinese hamsters, canines, and at the same time we have developed them from the human beings, from the human cervix, and which is the HeLa cell line, and the human lung, which is the W38 cell lines. So cell lines have been developed from a large number of the animals, and to culture the viruses